my dear little champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 days, and today we are going to do the second topic from biotechnology principles and variation, which is isolation and amplification of DNA, my little champions. I thought I should not be very complicating it very much, so I have not included the restriction endonucleases enzymes and that part over here. So if you are looking for that, I'll tell you, you should be waiting for the next video which will be coming along the way. Right? So today this is just isolation and amplification of DNA. I'll be talking about PCR and I'll be talking about what all enzymes are needed for isolation and things like that. Yeah, let me go ahead with the topic. And what all did we cover in the previous session? We spoke about what does biotechnology deal with, techniques that are involved. I wrote it twice, the origin of replication, the first recombinant DNA, so many questions have come from that. So if you want the notes for all those things, you can join our WhatsApp channel, my little chutkus. And if you're done with that, let's see the number of questions. It was 5 in the previous year, so 5 into 4, 20 marks from this chapter, not something that is avoidable. Now, remember giving something like this as a homework. Let's see who all got it correct. So, the sequence that controls the copy number of linked DNA in the vector is termed as. So I told you the vector, for example, this is a plasmid. Okay, and I have put the DNA of my interest over here, which is known as the gene of interest. Now I put it here because I wanted it to replicate, increase its number, and at last undergo the process of transcription, translation, and produce the protein which I wanted. Now suppose it is not undergoing replication. If it's not undergoing replication, okay, transcription, translation happened, suppose. I just got one protein, but I need much amount of protein, right? So replication is important because if replication happens, then I'll get multiple copies of the DNA that will code for multiple proteins. So that replication will only happen if I have a sequence which is known as origin of replication. With love, I call it ORI, O-R-I. Okay, what do I call it? O-R-I site. So if this site is there, then the DNA polymerase can come and attach and undergo replication. If it's not there, then this process won't be happening. So the copy numbers are basically decided. Remember always replication and copy number. I'll write it somewhere. Okay, ORI decides the replication. And the copy number. Works? Cool? This becomes the answer. This is a question that came in NEET 2020. Cool? Chalo. Let's see today's topic, that is isolation of DNA. How do you think isolation of DNA happens? Let's read the NCERT and think, and let's see what all do we understand from it. Chalo. Recall the nucleic acid is the genetic material of all organisms without exceptions. Yes. Do you have anything else as a genetic material? No. I have DNA and I hope that you also have DNA. In some organism you have RNA as genetic material, but it's DNA and RNA both coming under nucleic acid category. DNA is deoxyribonucleic acid, RNA is ribonucleic acid. Both of them are nucleic acids. Okay, done. In majority of organisms this is deoxyribonucleic acid. Agreed? Okay, in majority of them we have DNA as genetic material because that is more stable than RNA. In order to cut the DNA with restriction enzymes, it needs to be in the pure form, free from other macromolecules. Since the DNA is enclosed within the membranes, we have to break the cell open to release the DNA, along with the macromolecules such as RNA, proteins, polysaccharide, and also lipids. What? So basically, first step that I have over here is, okay, step 1 is breaking the cell, okay, because we want the DNA, so we want to break the cell. Now children, the cells could be different. For example, I am taking a cell, plant cells DNA, okay. There is a plant cells DNA over here. This is a plant cell. So plant cell has an outermost layer which is formed of cellulose. What is this layer? It is known as cell wall. So to break this cellulose, I need an enzyme. What do you call it? Cellulase. Okay, I need to break it, so I need to have cellulase. Similarly, suppose this is I need the DNA of fungi. Suppose I need the DNA of a fungi. Now fungi again has something that is known as cell wall. Again it has something that is known as cell wall. but the cell wall is made up of something else which is known as chitin. Okay, so to break chitin down, what do I need? I need another enzyme, chitinase. Okay, I need another enzyme, that is chitinase. Suppose now there is another thing, that is bacteria. Okay, it's a bacteria, it's a prokaryote. Prokaryote also have cell wall. They also have cell wall, and their cell wall is made up of something that is known as peptidoglycan. Now, peptidoglycan could be broken down with the help of an enzyme which is known as, okay, I should change the color, which is known as lysozyme. Remember, you have studied somewhere lysozyme. Basically, cellulose is broken down by cellulase, chitinase, and you have lysozyme. So lysozyme are there in our tears, the saliva and things like that. So that's why we say that tears have an excellent way of protection. Nothing can get inside. Okay, so basically not tears, eyes have a very good protection. Why? Because tears can be produced, and this tears will be rich in one enzyme, that is lysozyme, which will protect from all the bacteria. It will kill all the bacteria's things. Okay, cell wall is cleaved with the help of that. So first problem is solved. Okay, how do you get inside the cell? That is solved. Going ahead. Okay, let's come back. Yeah, let me see if I am, no, chalo, ayyo. Now there is step 2 where I got inside. Now inside I found that there are proteins. Okay, there are proteins. Now what else? There will be lipids in there. Sometimes there will be other nucleic acid also. RNA is also there. But what do I need? I need DNA. So while I am isolating the DNA, this is the guy that I want. So this is wanted, but I don't want them, so I kill them. I want it alive, but I don't want them. So I have to treat the cell with enzymes. For killing proteins, I need proteases. For killing RNA, we need RNase. For killing the lipids, I need lipase. You get it? So I took proteases for killing proteins, RNases for breaking down RNA, and lipases for breaking down the lipids, such that I have my guy, that is what? The DNA over here. Now, if someone says, I have one more enzyme, DNase. If that is put, then the DNA will also be gone. Okay, so we should not be putting that. So DNA is something that we need. It should be kept like that. Fine. So that is done. Now what? So I have took the cell, I have killed it. Okay, or basically I have broken down, not killing it, basically I have broken down the cell wall in somehow. I have got inside, I have broken down the proteins, RNA, lipids, and I have taken the DNA that I have over here. The next process is basically I have to separate the DNA from all the content. So I use a centrifuge machine. I have a test tube like this where I put all the content. Okay, and in a centrifuge machine, you know, it uses a centrifugal force to rotate things like this. Okay, goes really fast. At this point of time, the DNA which is there, okay, the DNA which is there, it will come settle down at the bottom. It will come precipitate. It will come precipitate. Okay. Over here, we will read it once and then we'll go ahead. Fine. So, yeah. So basically, I should be getting them free from other macromolecules such as basically lipids and things like that. So since the DNA is enclosed within the membranes, we have to break the cell open to release DNA along with the other micromolecules such as RNA, proteins, polysaccharides, and also lipids. Polysaccharides as in carbohydrates are also there. This can be achieved by treating the bacterial cell or plant cell or animal tissue with enzymes such as lysozyme for bacteria, cellulase for plant cell, and chitinase for fungus. You know that genes are located on long molecules of DNA intervened with proteins such as histones. The RNA can be removed by treatment treatment with ribonucleases, that is RNases. Okay, ribonuclease, RNase is same thing, whereas protein can be removed by treatment with protease. Other molecules can be removed by appropriate treatments, and purified DNA ultimately precipitates out after addition of chilled ethanol. This can be seen as a collection of fine threads of suspension. So basically, this will be done. Okay, this separation will be happening. One very important thing that I left is you have to add chilled ethanol, my dear children. When you add chilled ethanol, at that point of time the DNA will be precipitating down. Okay, if you are not doing that, that's a big problem. So that is also very much needed. And tada, we have got the DNA over here. And at the end, this picture, if you notice, they have written DNA separates out and can be removed by spooling. So this process of taking in, there's a thing like this, okay, which is used to collect the DNA like this. This is known as spooling. So the last step that comes over here is, children, spooling. Okay, DNA will be precipitating when chilled ethanol is put. This is also very much important. So this is how isolation of DNA works. Now I have taken down the DNA. Now what has to be done, children? Taken down the DNA. The next thing that I have to do is basically the important things that I have. Okay, genes which is of my interest, I have to cut it down with the help of restriction enzymes, restriction endonucleases. So next thing that I am going to do is cut the DNA with the help of restriction enzymes or restriction endonucleases. These will be cutting the DNA into and will be giving the gene of interest to me. Okay, that step we will be making a separate video for that, and I will be explaining you that really well. As of now, this is the thing that we have in hand. Okay, now going ahead. After cutting the DNA, so the cutting of the DNA by restriction endonucleases results in the fragments of DNA. You will be having small fragments of DNA. These fragments can be separated by a technique that is known as gel electrophoresis. Okay, what do you call this process as? You call this process as gel electrophoresis, where what I am doing, children, you look at this very carefully. Okay, so basically I have something like this. Okay, and suppose this is cathode, which is positively charged, and over there you have anode, which is negatively charged. Okay, there are certain wells over here, small wells. Fine, and these guys, they are connected to a power supply as well. Now we have DNA fragments, so DNA got cut. Our DNA which was there, the long DNA, it got cut with the help of the restriction endonucleases, the ones that is responsible for cutting. What do you call them? In other language, molecular scissors. What do you call them? Molecular scissors, children. Now according to the length and all, I have to differentiate them, all of them. So what do? In this particular thing, I am putting a gel which is known as agarose gel, and I obtained this from seaweed. I get it from seaweed, that is the red algae that we have. Okay, now this agarose gel is there. The wells are also there. Wells as in there are small holes near it. Okay, in this particular region, where I am going to put the DNA. Okay, now what will happen, children? Basically, when I turn the power supply on, just a second, I have to bring out certain changes over here. So now it is correct. Cations are positive, so this is anode itself. Anions are negative, but over here anode is positive. A little bit of chemistry mistakes, please forgive me. So this is positive and this is this is positive and the cathode is negative, and the cathode is negative. I hope I'm correct. Okay, but I really want to check, so I'll just check it and I'll come. Just give me a second. Yes, I was right. So now listen to me carefully. The DNA that I have is negatively charged, my dear children. DNA that we have is negatively charged because of the amino acids which is present over there in high amount. Okay, so they are negatively charged thing. This is positive end, this is negative end. So when electric supply is provided, what will happen, children? These guys will start moving over there, will start moving towards the anode. Now the guys which are smaller in size, they will be reaching first. They will be like, yeah, we reached it, we reached the point. And over here, the ones which are larger in size, they will be coming out slowly. So these were the larger ones, these are the medium ones. Okay, they will be coming slowly. So what do you understand from this? Basically, the DNA fragments that you see would be separated according to its size, okay. So it is smaller size, they will go ahead, the larger ones stay behind, but all of them, they are moving towards the anode. Why they are moving towards the anode? Because children, they are negatively charged, negative and positive charge. So basically the opposites, they attract, you know that, and if they are the same type, they will repel. So it will move from this end, because this is also negative end, it will go to that end, that is positive end. And over here, what do you find? The smaller fragments moves first, okay, and the larger fragments moves slowly, okay. But now the problem is that I cannot see which one is smaller, which one is darker, or which one is larger, not darker, which one is larger, which one is smaller. They are not visible in this particular thing. And this whole procedure, what do you call this, children? This whole procedure is known as gel electrophoresis. It is known as gel electrophoresis because you are using a gel for separation of the DNA content over there. Now they are not visible. So what do? I can actually treat it with something that is known as, ethidium bromide. What is it, children? It's a chemical, it's a dye, okay. What do you call it? Ethidium bromide. Now when they go and attach, they get inside the DNA, what will happen? It gives a orangish color. But still I cannot see it. Even if I put ethidium bromide, I cannot still see with the naked eyes like this if it is natural light that is being put. I need some different form of light, okay. I need some different form of light, which is known as UV radiations. So when UV radiations are put at this point of time, this ethidium bromide will be giving the orange color over here, my dear children. Okay. Now one more thing that you should be knowing is that ethidium bromide, what does it do is that they get inside the base pairs of DNA. So this is a DNA. You know there are base pairs, A goes with T, G with C. So it comes between the base pairs of the DNA and gets in there. So it is used in very less amount. If it is used in more amount, it can be really bad for the DNA. It can cleave the DNA off. Okay, it's not really safe for the DNA. So it is used in very minimal quantity, and also you know UV radiations are also not that great for the DNA. Anyhow, everything is used in a very limited proportion. That is how they are turned out to be visible. Okay, now this is known as gel electrophoresis. What all do you need to know is that you have a positive end, you have a negative end. The DNA will always move towards the positive end because they themselves are negatively charged. Now they move accordingly according to the fragment size. For example, the smaller fragments will be moving there very fast, the larger fragments will be moving back. Okay, they will be moving not moving back, they will be moving really slow. Now to view them, okay, you need to treat them with ethidium bromide. Ethidium bromide will give orange color, but when they are put under the UV radiation, at that point of time they will show up. And this whole process is known as gel electrophoresis. Now if you are also a person who get confused like I am with positive and negative things and like that, you can remember like this, okay. Cats have pos. That means cations are positive. Okay, cations, they are positive. So cations are positive, but cathode is negative. Similarly, anions are negative, but an anode is positive. You can remember like that. Okay, if you wish to. Remember, this is very much important. This is very much important. This is very much important over here. The agarose gel that we use, you get it from seaweed, that is also given in your NCERT. Okay, Works? And accordingly you can separate your fragments over there. And now these fragments, whichever I want, I can take them and then I can replicate it, okay, using a machine which is known as PCR, about which we are going to study next. But before that, let's complete reading the NCERT for this particular topic and then we'll go ahead. Remember, there is a thing, isolation. After isolation you have to cut it with restriction endonucleases. That second step will be done in the next class, after which separation has to be done. Separation I am teaching you over here, and then you're going to amplify the number of DNA. That is done with the help of PCR. Okay, so the cutting of DNA by restriction endonucleases results in the fragments of DNA. These fragments can be separated by a technique that is known as gel electrophoresis. Since DNA fragments are negatively charged molecules, they can be separated by forcing them to move towards the anode under an electric field through the medium or matrix. Nowadays, the most commonly used matrix is agarose, which is a natural polymer extracted from seaweed. So it is naturally occurring thing and it is extracted from seaweed children. It's not a synthetic thing. The DNA fragments separate according to their size through sieving effect provided by the agarose gel. Now this is also very much important. Please remember, the DNA which moves around, it is not moving on the surface. It does not move on the surface. Rather than that, if this is agarose gel and I am DNA, this is agarose gel, I am DNA, I'm going to get inside and then move here. So there is swaying effect that is being shown. Swaying as in it moves like this, you know. How does a swaying machine sway it? So basically it gets inside. Similarly over here also the DNA is also moving inside the agarose gel, and this is an important question that has come in NEET. Okay, how did I forget it? So this is an important thing. Now hence the smaller the fragment size, the farther it will move. The farther it will be moving. Now the separated DNA fragments can be visualized only after staining their DNA with a compound that is known as ethidium bromide, followed by exposure to UV radiation. You cannot see pure DNA fragments in the visible light and without staining. Okay. You can see bright orange colored bands on DNA in ethidium bromide stained gel exposed to the UV light. So UV light is also important. The separated bands of DNA are cut out from the agarose gel, and the separated bands which are there is cut out and extracted from the gel piece. Okay, this is also important. This process you call it as elution. I'm writing it here. Removal of DNA from the gel is known as elution. Works? Okay, chalo. Now going ahead. The DNA fragments purified in this way are used in constructing recombinant DNA by joining them with cloning vectors. Now the next step will be cloning vectors. No, the next step won't be cloning vectors. Basically I want to increase their number first and then I will be putting it into vectors. Okay, so for that we need an enzyme. For that we need a machine which is known as PCR. I hope this is exactly clear to you children. Now we can go ahead, right? Okay, let's start with PCR. This is one very interesting thing. So the machine which is responsible for making the multiple copies, you call it polymer chain reaction, and the reaction also you call it polymer chain reaction. So PCR stands for polymerase chain reaction. In this reaction, multiple copies of the gene, that is DNA, of interest is synthesized in vitro using sets of primers. We will come to that. And the enzyme DNA polymerase, which is why polymerase chain reaction term has come. Okay. So what is the first step? Let's see. The first step that we have is denaturation. Okay, what exactly happens in denaturation? So you have the DNA strand, okay, which have bonds between them, A, T, all of them, they have bonds over here, you know that. So the first step is done in almost 90 degrees Celsius to 92 degrees Celsius over here. Okay, it's too high temperature. So with this temperature, what happens children? The DNA strands, it gets separated. The DNA strands, it gets separated, and this is known as denaturation. This is known as denaturation. Okay. Second thing that we have over here is annealing. Second step is annealing, and this is done Okay, what do you mean by annealing? So you have the denatured DNA, so I am putting it over here. This is the denatured DNA. Suppose it has a code that is A, T, G, C over here. A, okay, and the strands are going like this. This is 5 dash, this is 3 dash. The opposite DNA strand will be opposite, so it will be 5 dash, and this will be 3 dash end, right? Okay, now over here again I am putting the same thing, A, T, G, C. Okay, anyways, I've just put it like that. Annealing means you want to make a copy of the DNA strand. So this DNA strand is there, I want to make a copy of it. I want to make a copy of this strand also. And I know the strand sequence. I know this strand sequence. So what do? I make a primer out of it. Primer should be having complementary strand. So here the primer will be having, suppose this is the primer, which is in 5 dash through 3 dash direction. The primer will be having A is here, so it will be T here. T is here, so A will be here. G is here, so C will be here, and C is here, so G will be here. This is the primer that I have created. Now mixing it with the solution that has the denatured DNA. What will happen? All these primers will go attach over here. So this is the 5 dash end. Over here I have T, A, G, C, and this is the 3 dash end I have. Similarly, this primer will also come over here. Same. The 5 dash end, it will be T, here it will be A, here it will be C, and it will be G. Okay, in this format it will come and attach. Why am I doing this? Why do you think annealing is done? Because, you know, for copying the DNA, you need an enzyme which is known as DNA polymerase. You need an enzyme which is known as DNA polymerase, which does its working in 5 dash to 3 dash end direction, which forms thing which makes a new DNA in 5 dash to 3 dash direction. So if this is 3 dash, then opposite one will be 5 dash, so from here only it will start. But for that it needs the first nucleotide. It needs the first nucleotide, then only, or first or second nucleotide, then only it can continue the reaction. So it says without the help of primers I cannot do it. So these primers are synthetically created, and it is put over there so that the primers will go and bind to the complementary strand. Now depending on whatever strand is, for example it is G, T, A, C, whatever is there, depending on it, now the DNA polymerase will come. DNA polymerase will come and extend this. For example, G is here, G is here, so it will be C over here, it will be A over here, it will be T over here. In this way it will go on extending in the 5 dash to 3 dash direction, but it needs the primer initially because of the bonds that is needed over there. Okay, so that is why we need primer, and the attachment of primer over here. The attachment of primer over here, it is known as annealing, and this needs a temperature of around 55 degrees Celsius. So it has become lower. The temperature has lowered over here in the case of annealing. Please remember, you need what, children? DNA polymerase over here for the extension. Okay, but extension I have not come to that. I am talking about annealing. For annealing, what do you need? You need a primer. So as of now, I am just going to erase this. As of now, I am just going to erase this because I am not talking about elongation. I have just spoke about annealing, and for annealing what do you need, children? You need a primer. And what is a primer, children? It is just an oligonucleotide, just a strand of oligonucleotide. So when I say there is A, C, this is a nucleotide. Okay, how do I say, why do I say this is a nucleotide? Basically you know what is a nucleotide? I'll show you. If you have not studied with me previously in molecular biology, I'll show you how does it look. So this is a sugar which is known as ribose sugar, deoxyribose sugar actually. This is first carbon, second carbon, third carbon, fourth carbon, fifth carbon is like this, and fifth carbon has a phosphate group attached to it. Okay, first carbon has a nitrogen base attached to it, that is, it can be A, it can be T, whatever is it. So this is a nucleotide. The thing that you see over here, that is A and T, it is the extending part of it, which is the nitrogen base. So all these things that you see are cute little nucleotides coming together, which have one base each. So here it has G base, that is guanine sugar. Okay, that is guanine over here, or it has not guanine sugar, basically guanine over there, nitrogen base, or it can have cytosine, or it can have adenine, or it can have thymine. Depending on it, you have different types of nucleotides. So I am saying primer is what? An oligonucleotide. Oligo as in it is made up of more than one nucleotide. So it has multiple nucleotides coming together, different nucleotides or same nucleotides coming together over here. So primer is an oligonucleotide which comes and attaches over here, and this process is known as annealing, which needs a temperature almost around this. It's not given in the NCERT. Okay. Now the next process will be if this is the second process, the next process is third process is known as extension. Okay, the next process will be known as extension, where what is happening, my dear children, this particular strand that you see, it is going to extend. This particular strand that you see is going to extend with the help of what, children? The DNA polymerase. And now, as I said, it will go extend in this direction. It will be C over here, it will be A over here, it will be T over here. This way it will keep on extending. So this is extending in 5 dash through 3 dash direction. This is known as extension. Works? Cool? Okay. What is the temperature? 70 degrees Celsius. So if you see, first it was 90 to 92 degrees Celsius, then it came down over here, then it has now again gone high for extension process to happen. And in this way you are creating, so from here I am getting one strand was like this, I am getting another strand which is coiled over here, another strand which is coiled over here. So from here I got 2 copies. Get it? And this process keeps on repeating. Now the thing is, which is that enzyme which would be able to break down the hydrogen bonds at this particular temperature? Which is that enzyme which would be able to break down the hydrogen bonds at this particular temperature, like this? So bases, if you see, this base is basically they are attached. A and T has 2 hydrogen bonds, G and C has 3 hydrogen bonds. So there should be some enzyme that should be breaking down in the process of denaturation, and this enzyme, I am telling you, is an enzyme which is known as Taq polymerase. What do you call it? Taq polymerase. Okay, we'll come to that, but before that we should be reading the NCERT. Chalo. So the enzyme extends the primers using the nucleotides provided in the reaction and the genomic DNA as a template. Over here, one more thing I want to add up is that where do you get the nucleotides from? This is also put in the mixture. These nucleotides which are coming in the extension, that is also there in the mixture, they will come and just attach depending on the complementary base pair. Okay. Yeah. If the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately billion times, that is 1 billion copies are made. Such repeated amplification is achieved by the use of thermostable DNA polymerase. Okay, thermostable DNA polymerase isolated from a bacterium Thermus aquaticus, which remain active during high temperature, induce denaturation of double-stranded DNA as well. Okay, so I said that this everything, where is it? Everything is possible because of an enzyme, because of a different type of enzyme that we have, which is known as Taq polymerase, which is responsible for replication, which is responsible for breakdown, denaturation and everything. Okay, so where do you get it from? You get it from a thermostable bacterium. You get it from a thermostable bacterium, my dear children, which can withstand high temperature, and its name is, remember the name, it's very much important, Thermus aquaticus. Thermus aquaticus. So T from here, AQ from here, that is why you have Taq polymerase. So this Taq polymerase is a type of DNA polymerase which is used for replication as well as denaturation of the DNA strand. Works? Thermus aquaticus is a very thermostable bacterium. So obviously thermostable bacterium as in it is able to withstand high temperature. So it's okay for it, it's totally fine for having 90 degree, 92 degrees Celsius or 55 or 70, whatever, it's totally cool with it. Okay. So this is about your PCR, my dear children. You get so many copies, and the amplified fragment, if desired, can now be used to ligate with a vector for further cloning. Next step will be cloning with a vector. So you have a vector, you put it over there and you join it with the help of DNA ligase. That is the next step, about which we are going to study in the next class. Okay. First we will study restriction endonucleases, and then only we will go on with the cloning part. Okay. So here you had the DNA. With the help of heat, you denatured it. Okay, now you have 2 separate strands. Now with the primers, you are coming in annealing this DNA polymerase, that is Taq polymerase plus deoxynucleotides. You are using deoxynucleotides, the one that does not have OH group over here. So usually, I'll tell you one more thing. So usually in the case of ribose sugar, no, that is in the case of RNA, there's an OH group over here. But here the OH group is not there. Here this is just H, which is why it is known as deoxy. So you need what all, children? You need Taq polymerase, and also you need nucleotides. You also need deoxynucleotides. You need Taq polymerase to extend it. Okay, that's the enzyme. And this nucleotide should also be coming. So these are known as deoxynucleotides. Works? Okay. So then extension is done and then amplified 30 cycles, it will be giving you 1 billion copies of DNA. I hope this is done. Okay, so we spoke about isolation of DNA, we spoke about gel electrophoresis, and we also spoke about amplification of DNA with the help of a machine which is known as PCR, polymerase chain reaction. And I hope you got each and everything that I explained over here. Now we can try questions from this topic, so you'll get an idea of how questions are being framed. The first question that we have is given below. Okay, I'll be telling the answers for the same later on. You have to try the questions by yourself first. Okay, so given below are 4 statements pertaining to separation of DNA fragments using gel electrophoresis. Identify the correct statements. You have to identify the correct statement. Okay, DNA is negatively charged molecule, and so it is loaded on the gel towards the anode terminal. Is it put towards the anode terminal? What is anode terminal? If you remember, anions are negative but anode is positive. So can you put it in the positive side? Think about it. DNA fragments travel along the surface of the gel whose concentration does not affect movement of DNA. Do we think that the DNA is put on the surface of the gel? Again, this is something that I explained. Smaller the size of DNA fragment, larger is the distance it travels through it. The smaller the DNA, the larger it goes, the larger distances it travels. That is said. That is said. Pure DNA can be visualized directly by exposing UV radiation. So you have DNA, just put it under UV radiation and you will be able to see it. You have to identify the incorrect statements, not the correct statements. Okay, so you can choose from the option. This came in 2019. Next question. It will be really good if you tell me the answers now. I'll be really happy seeing you try. Now, DNA precipitation out of a mixture of biomolecules can be achieved by treatment with. How can you separate them out? DNA. Chilled chloroform, isopropanol, chilled ethanol, and methanol at room temperature. This came in NEET 2019 again. Third question. Which of the following is not correctly matched for the organism and its cell wall degrading enzyme? Algae, methylase, fungi, chitinase, bacteria, lysozyme, plant cell cellulase. NEET 2013. Simple question that we have got to understand. So simple, right? You should be trying it out. Next, fourth. DNA fragments are negatively charged, neutral, either positively or negatively charged depending on their size. Positively charged. NEET 2017 question. You see, you can score easily in these type of questions. Now, in gel electrophoresis, separated DNA fragments can be visualized with the help of what? Acetocarmine in bright blue light, ethidium bromide in UV light, acetocarmine in UV radiation, ethidium bromide in infrared radiation. NEET 2020 question, not too far, it is a recent question. Okay, this is the fifth question. Now, let's try solving it by ourselves. So, over here, given below are the 4 statements. DNA is negatively charged, agreed. So it is loaded on the gel towards the anode terminal, no children. It is towards the cathode terminal. It is put in the negative side only. This is cathode. Negative side is cathode. From here, the negatively charged will repel and will move towards the positive end, that is the anode. So this is wrong. So first is wrong anyway. So we have to find the incorrect one. So this is one incorrect thing. DNA fragments travel along the surface of the gel. No, it has a swing effect. So it travels through the gel within inside. So that is also wrong. First and second is wrong. Smaller the size of DNA fragments, larger is the distance it travels. Smaller the size, yes. Larger the distance it travels, it is true. So wherever you see 3 option, you cut it. So this is 3 option cut, 3 option cut. 3 option cut. What you're left with? You're left with this. Chalo, let's see the fourth thing. Pure DNA can be visualized directly by exposing UV radiation. No children, you need ethidium bromide, then only it will be visible. So this becomes the correct answer. These 3 points are wrong. This is wrong, this is wrong, this is wrong. Okay, DNA precipitation out of a mixture of biomolecules can be achieved by the treatment with. DNA precipitation, how can it be achieved? It can be achieved by the treatment with what, children? Chilled ethanol. NEET 2019 question. Going ahead, which of the following is not correctly matched for the organism and its cell wall degrading enzyme? So fungi, you know, it is made up of chitin, so chitin is correct. Bacteria, you know, peptidoglycan, so lysozyme is correct. Plant cell cellulase is there. So algae by methylase, that is wrong. So this becomes the answer. Okay, DNA fragments are negatively charged, no questions regarding that, and they move towards the positive side. Now, in gel electrophoresis, separated DNA fragments can be visualized with the help of something that is known as ethidium bromide, which gives the bright orange color, children, and it can be viewed only under UV radiation. With the normal light, it cannot be visible. So, ethidium bromide in UV radiation, this becomes the answer. Cool? going ahead, one question for your homework, my dear little champions. Okay, that is, during the purification process for recombinant DNA technology, addition of chilled ethanol precipitates out what? So when you are doing the purification process at this point of time, you are putting chilled ethanol. What does it do? What does it precipitate? This came in NEET 2023, very recent. So tell me the answer for that in the comment section, and tell me if you are liking the videos, if you are liking the content, not liking the content, liking the content, and everything is good. And please be rest assured that you're going to ace the exam, my dear children. You just have to give your best, and I know you will be giving your best. If you think that this particular video was really good, do not forget to subscribe to our little channel as well. Yeah, so bye bye, take care.